You're a detective on the case for a recent theft. Luckily, the culprit left behind some hair at the scene of the crime, and 3 suspects are currently being detained. You just need to send the hair over to the forensics lab and hope the DNA matches one of the suspects. But wait a minute, what does it actually mean for DNA to match? DNA is invisible to the naked eye, so how can we tell if 2 samples are the same? In cases like this, scientists can use a method called genetic fingerprinting. But before we get into that, let's talk about DNA. DNA is made up of nucleotide chains, with each nucleotide containing a negative phosphate group and a sugar group that make up the backbone of DNA's double helix. It also has 1 of 4 nitrogenous bases: adenine, guanine, thymine, and cytosine. In genetic fingerprinting, a strand of DNA is divided into smaller pieces using restriction enzymes before they are separated with gel electrophoresis. Restriction enzymes are like really picky ninjas, as a select enzyme will only cut the DNA between specific base pairs. For example, the restriction enzyme EcoR1 only cleaves DNA between the base pairs adenine and guanine. So in this strand of DNA, the restriction enzyme would cut here, here, and here. Now it's time to get electrical with gel electrophoresis. This is a method that separates molecules based on their size. Here is a gel electrophoresis machine with a block of agarose gel in the center. On the left, there are wells, which is where the fragments of DNA will be placed. When an electrical charge is running through the gel, the side with the wells will be negatively charged, while the opposite side will be positively charged. Because of DNA's negatively charged backbone, its attraction to the positive end of the machine will cause the DNA to move through the porous gel. When the original sample of DNA is initially divided, not all of the fragments will be the same length. Some will be longer than others, and these strands will have a higher molecular weight. This means that shorter strands of DNA will move through the gel much faster than longer strands. Let's place the DNA sample from the hair we found at the crime scene next to the DNA of the 3 suspects. After running the gel electrophoresis machine, we end up with a pattern of DNA bands that looks like this, with the larger bands staying close to the well, while the smaller pieces are further away. This pattern is what we call a genetic fingerprint, and would you look at that? We've got a match. DNA fingerprinting and gel electrophoresis can also be used in paternity and maternity testing, as well as to analyze relatedness between species. The study of genetics and biotechnology are fields that are constantly evolving, but for now, you have a thief to arrest, detective. Case closed. Thank you for watching. This video was created by Jacqueline Cheung, and the sources I used are listed down below.